event. It's now my pleasure to introduce our next speaker, Professor Colin Pouton. Colin is a professor of pharmaceutical biology at Monash University with extensive research experience across various aspects of drug discovery, drug delivery, and development. Since early 2020, Colin has been using his experience in nucleic acid delivery to design and deliver candidate mRNA COVID-19 vaccines. Today, Colin will be discussing the exciting milestone that he and his research group are currently working towards, taking the first Australian mRNA vaccine to phase 1 clinical trials. So with that, welcome to BioForum, Professor Pouton.
Thank you very much, Jeff, and I'm delighted to be here. I'm going to start by just giving an overview of why we're so fascinated and interested in the potential of mRNA technology, and then I'll go into a bit more detail about what we're doing precisely with our COVID-19 vaccine development. Next slide. So, as Minister Pulford said, the research around mRNA delivery has been going on for a number of years, but in many ways the COVID-19 pandemic has really given the opportunity to exemplify this technology. And the huge amount of funding that was put into, the public funding that was put into development of the vaccines that are now approved is something which is really a remarkable opportunity and was seen as a remarkable opportunity as soon as the pandemic started. I think those of us working in the field knew that this is an opportunity to test mRNA technology, but I don't think anyone really believed, well, not believed, but anyone really could have hoped for such remarkably good results. And so that is exemplification of the technology really opens the door to all sorts of other opportunities. So the reason we're so excited about mRNA is that it's a process which is cell-free, and from a pharmaceutical point of view, I'm a pharmaceutical scientist, it represents a form of therapy which is potentially easier to deal with, easier to produce and manufacture, and also a lot safer than other approaches to genetic forms of therapy. So there are still, though, lots of opportunities for improving the design of mRNA and also the delivery. And in many respects, it's the delivery to different parts of the body, to appropriate places, which has really held back the development of mRNA. And you've probably heard about the way in which mRNA is formulated in lipid nanoparticles. That delivery technology has really enabled these vaccines to work. It's a very important component of the whole thing. And the coming together of the mRNA design and the delivery has really made this, these vaccines possible. So I'll mention a bit more about that later. In terms of the general idea, the mRNA technology has got some really clear advantages, which I've summarized in those dot points. So certainly one of the things about it, and the diagram, a schematic diagram of what we're dealing with is shown in that diagram in the left corner there. It's a pure molecule which we can purify using pharmaceutical techniques. And it's a large molecule, but it's one that we can produce and characterize very cleanly. And this leads to QC, quality control, and quality assurance advantages in pharmaceutical manufacture. Another very important point is the safety aspect, because this is a form of gene therapy, but it's not a form of gene therapy that can interact with with the genomes themselves. So it's a genetic code which comes from DNA to RNA, but it's not something which can be inserted into the DNA genome. And that makes it very safe in relation to other forms of gene therapy. And we say, therefore, there's no possibility of insertional mutagenesis. Of course, the fact that we're working with genetic sequences and we sequence DNA, we can make RNA quickly, meant that you could produce a product for trialing as a vaccine candidate very, very quickly. And so, well, Moderna say that they produced their first candidate vaccines for COVID within days of hearing the sequence, of having the publication of the sequence available. And they reached the first phase 1 clinical study in humans in a little over 2 months, which is really, it's quite remarkable, unprecedented speed. And of course, that means that this kind of technology is ideal for responding to an emerging outbreak from a virus, such as in this pandemic. The other thing that's really important is because we can modify the sequence very easily, it means we can respond to changes. So as the virus mutates, as we're seeing now with the variants emerging, we can respond to that and produce vaccines that are tailored to meet the new variants. And the product itself looks very similar to the original. If you imagine the product we're dealing with now, which is a Wuhan sequence, if we want to change that to a product which targets a variant, the product itself, the mRNA and the delivery system look very similar. So safety aspects are easy to deal with. There are some other issues that are important too, and that is that if we have a series of variants emerging as they are now, potentially we can give a cocktail of sequences to immunize against a whole variety of variants. And that's something which is very much on the agenda at the moment. The companies involved in the technology are working on this, and so are we, to ask the question, what's the most efficient way of vaccinating against a broad number of variants? And there are some, at the bottom, the 2 bullet points I've put there, are a couple of things that indicate the level of, that it's not a single product, that there are opportunities for developing different forms of RNA. One of them is the so-called self-amplifying RNA, and that means that you can reduce the dose quite substantially from what we're giving now to something which is perhaps 100 times lower. And there are some self-amplifying RNA vaccines in the clinic for COVID-19. We have to wait and see how well they perform, but they've got a lot of potential in reducing the dose and therefore reducing the amount of material you have to make to vaccinate the population. And also I should note that we're dealing with these vaccines with materials that are chemically modified already. So they're not native RNA. They're actually slight variations in the structure of the RNA, of some of the components of the RNA, and that changes their response. It changes the immune response to these materials. So there are a number of options there in relation to the design, and it affects the immune response in different ways. So there's a lot still to explore in terms of optimizing the vaccine. The next slide. So I'll tell you a bit more about what we're doing and how our vaccine that we're developing in Victoria differs from the existing vaccines. So the diagram on the right is a schematic diagram in figure A of the sequence of the spike protein in the coronavirus. So just showing the different regions of the spike protein. And in figure B, that's a structural study of what the coronavirus spike protein looks like. It's actually three identical proteins sort of intertwined together into what we call a trimer. And in the green part of that structural diagram at the top, you can see what we call the receptor binding domain. In figure A, you can see that part of the protein in green with the label RBD, receptor binding domain. That's the part of the spike protein that actually binds to a protein in our cells, the so-called ACE2 protein, which acts as a receptor for the virus. So it's against that RBD that we need to raise antibodies. By blocking the RBD and blocking its binding to the ACE2 protein, we can neutralize the virus and stop it infecting our cells. And that's the main strategy behind all of the vaccines that are in use at the moment. So turning to the bullet points on the left, the difference between our protein and the vaccines that our vaccine and the vaccines that are already in use is that the current vaccines use the whole spike protein. So they're delivering mRNA, which presents the whole spike protein. And what that does is to raise antibodies against not just the RBD, but also against other parts of the protein. You can see from the diagram that the RBD is actually quite a small part of the whole protein. It's around 15% of the whole protein. So what we're trying to do is to, instead of vaccinating with the whole spike protein, we're vaccinating with just the RBD component of the vaccine, of the protein. And what that means is that we can hopefully raise antibodies specifically against the receptor binding domain. That's particularly important in relation to the variants, particularly in patients who've already been vaccinated with the current vaccines, because they're using the Wuhan spike. Now, what that means is that if we've been vaccinated with the Wuhan spike, and yet we're trying to boost against a new variant, such as the Delta or the Beta variant, we need to raise antibodies against that modified RBD. The problem that we have is that if vaccines are just modifications of the whole spike protein, then the patient is primed to produce antibodies against other regions of the protein, and they don't necessarily produce new antibodies against the variant RBD part. So by using the RBD, we're hoping that we'll be able to, if you like, bias the immune system towards the new antibodies that we need to fight off the particular variant of interest. And in relation to the elderly people who suffer badly from COVID-19, there's some relationship there between the immune response to the virus and the immune response to the vaccine. It may well be that patients who suffer really badly from COVID-19 are just raising non-neutralizing antibodies because they've been primed against a coronavirus spike for a different virus, a virus that resembles SARS-CoV-2, but isn't actually SARS-CoV-2, has a similar spike, and therefore they're primed to produce non-neutralizing antibodies. And that's one of the theories that immunologists are looking at and trying to find ways around. Next slide, please. So a bit more detail here on what we're trying to do and what the variants are. There's two diagrams here. The diagram on the left is, again, a sort of schematic of the whole spike protein, but in panel B is the amino acid sequence, and those letters are the single amino acid, the codes for the single amino acids in the protein. So it's a sequence of a couple of hundred amino acids that make up the RBD. But what you can see is that, and it's in light blue in this diagram in A and in C, but there's also a region within that RBD, which is shown in red, which is actually the part that really binds to the ACE2 protein, which is shown in green in the diagram on the right. So that is called the receptor binding motif. And that receptor binding motif is the region that's most important in relation to different variants that are emerging. So let's take the next slide. If you look in more detail at those sequences, and what's shown here in this diagram is that RBD region again, and there are two sequences here shown. They're sort of aligned, they're aligned with each other, one on the top and one on the bottom in sort of in 4 rows. So on the top, we have the amino acid sequence of SARS-CoV-2, the coronavirus we're suffering from now. Underneath it is the amino acid sequence of the SARS virus, which caused the outbreak, didn't reach Australia, but caused the outbreak about 10 or 12 years ago. What you can see here is that this is the sequence for the RBD for these two viruses. You can see that in the blue regions, there's exactly the same amino acid between the COVID-19 SARS-CoV-2 virus and the other SARS virus. But in the regions that are shown in red, which are the receptor binding motif, you can see that there's no homology. What we say homology, in other words, the amino acid is not the same in that position. So there's a strong homology in some parts of the RBD, but not in the part which actually binds the receptor, shown in red. And what I've also done here on this diagram is I've superimposed, using those red arrows, the precise locations of the mutations that are causing the variants of concern. And you can see that, not surprisingly, these variants of concern are right in the receptor binding motif. But you can also see that whilst several of these have emerged, showing the arrow, where the arrows are, that there are multiple other places that potentially you could have mutations occurring. And part of the challenge going forward with this SARS-CoV-2 virus is going to be anticipating potential mutations that are poor, but that are bad and give us perhaps a more easily transmitted virus, or one which we're not protecting against with the Wuhan vaccine. So there will be a future development, second generation vaccines, if you like, which are looking at vaccinating better against these mutations, and also potentially anticipating the mutations that could occur, and potentially even worse potentially than the ones we've seen. Okay, next slide. So this shows you where we're at now with our current vaccine. What we're doing here is we're injecting our vaccine into mice. We're taking blood samples from the mice at different times, and this is actually after 2 doses. So typical prime and boost dose, which we hear a lot about. We're taking blood samples after 42 days in this case, and we're looking at how the mice respond, whether the mice serum can protect against infection. And there are 2 assays here. The one on the left is an assay which is done using cell culture. This was done in Damien Purcell's laboratory at the Doherty Institute. What they did is to take the serum from our mice and ask the question, can that serum protect some cells against infection by the virus? And Damien's lab can grow the virus and use the virus in an infection model in the high containment laboratories at the Doherty. Now, a large number on that scale, ID50, means good protection. It's actually about how many times you can dilute the serum and still get protection. And what we're showing in the black symbols is the level of protection afforded by the Wuhan version of the vaccine in individual mice. And there are different doses there. You can see what we're trying to do is to optimize the vaccine and work out what dose we need to give. So in the middle of that diagram, you can see 10 micrograms, 3 micrograms, 1 microgram. That's a purified sample of the Wuhan version of our vaccine. And in black is the neutralization of the Wuhan strain of the virus. In magenta is the neutralization against the beta variant, which is also called B1351. So you can see that the protection against beta variant is lower. It's about 10-fold difference. That's a logarithmic scale that we're looking at on the vertical axis. And so basically all of the vaccines we're giving are about 10-fold less neutralizing against beta. And that illustrates the problem that we're seeing with the vaccines. They're not quite as efficient against some of the variants. And actually, that beta variant is the worst in terms of its escape from the immune response caused by the Wuhan vaccines. And so we're going into a clinical trial with a version of our vaccine, which is a beta variant version. It's the best one to try because it's the one that escapes most. But obviously we're very interested in Delta as well. And there's less known about Delta, the way it's not quite as poorly protected against as the beta, but obviously there are other issues with Delta. It seems to produce more viruses and therefore is highly transmissible in the population. So very important to be able to protect against it. On the right-hand side of this diagram is an illustration that these vaccines protect mice from challenge with the virus. So that experiment on the right is an experiment that was done at Weehai in Mark Pellegrini's lab, where we took our mice down to Mark's lab and he inoculated the mice with what we call the N501Y mutant version of the virus. What's interesting about this is that the Wuhan virus doesn't infect mice, but the 501Y mutant does infect mice. And that was a mutant which actually arose in the first wave in Melbourne. It was isolated by colleagues at the Doherty, and now we can grow that virus and challenge mice with it. And it helps us to understand whether our vaccines are working. And in this particular experiment, the vaccines you're looking at are the same ones on the left-hand side. The mice have been vaccinated. We've taken them down to wee-high, and basically they're completely protected against challenge with the 501Y mutant, which is very encouraging. So next slide. So just before I wanted to acknowledge the team that have worked on this up to now. My team at MIPS, Harry, Estelle, Asaka and Tom have been very important in assembling the RNA and the delivery system. And certainly Peter at the Peter Doherty Institute, Damien Purcell has been very important in analysing the immune responses to our vaccinated mice. So next slide. I just wanted to spend a little bit of time talking about what the funding from mRNA Victoria is going to do for us. In particular, how we're going to be able to take our preclinical work into a clinical trial. So the important thing is there that we need to be able to manufacture using pharmaceutical conditions. So it needs to be a product which is suitable for human use and therefore has all the checks and balances required in a human pharmaceutical product. So I think we're getting into the concept of manufacturing and what's involved in manufacturing the virus. It's really 3 steps shown here, and that is that first of all we have to make some DNA, which is the template for making the RNA. You don't need very much DNA, and we're going to be making that in our laboratory and sending that DNA to a manufacturer to make RNA. We normally make it in our laboratory, but we're obviously not a pharmaceutical manufacturing entity. And we're sending, in order to get this done in time for a clinical trial, we're sending DNA to a company in Belgium that will make mRNA using that cell-free process. And at each of those 2 steps is a finished product. In a sense, you can freeze it, store it, ship it, and then you have to assemble it into a formulated product, which is step 3. So you have to incorporate the mRNA into, in the case of our formulations and the existing ones, the Moderna and the Pfizer vaccines, they're assembled into a lipid nanoparticle. Now that part of the process is something which we believe can be done in Victoria. And so the funding that we have from mRNA Victoria is going to help us to make a product which can then be brought back to Victoria and formulated at IDT Australia. So I wanted to say that there are these 3 separate processes. They actually could potentially be done in 3 different sites. And actually, that's exactly what Pfizer is doing to make the vaccine that we're using in Australia. They're making the DNA, the RNA in separate sites, and they're actually shipping RNA from one country to another to assemble the product as a lipid nanoparticle. So next slide. This is really the last slide, just summarizes what the funding that we have is going to be doing. So it's really going to help us to develop a product which is fit for human use to go into the phase 1 clinical trial. But in the process, obviously, one of the critical things we're trying to achieve is to introduce an understanding of the process locally in the manufacturing sector. So we're building capability and capacity for manufacturing in Victoria. And at the time, what we'll be doing is to use a piece of equipment which we're buying from a company in Canada called Precision Nanosystems. And I should say, actually, that our distributor for this piece of equipment, which is ATA, Pete Davis, I believe he's listening in. So thank you very much, Pete, for getting this piece of equipment sent across from Canada in time for us to make this product, because that was a key thing had to happen. And apparently the equipment is in Australia now, I'm delighted to say, and that is one of the miracles that we have to solve along the way to make this product in time for a clinical trial in good time. The other thing that we're doing, as I mentioned before, due to the sort of need to start this trial as soon as possible, is to have the mRNA made by a Belgian contract manufacturing organisation. Obviously, in the future, we're very keen for that capacity and capability to exist in Victoria. And that is one of the things that we'll be working on over the next period of time, to essentially put in place the steps to allow a company locally to be able to make the mRNA as well, and therefore do the whole process from start to finish. So we are going to formulate the product, filter it, fill it into vials at our local Victorian CMO, IDT Australia. And that process of assembling the whole product will be partly an opportunity for us to work with the local manufacturer to essentially tell them all we know about how to make this product, and hopefully their knowledge of pharmaceutical manufacturing will allow us to prepare for making other products in the future for clinical trials and for manufacture. Well, thank you very much. That's the end of my talk, and I'll hand back